Today we'll be learning about the metabolic signaling pathway of retinoic acid and why its proper function is critical during embryogenesis. What is retinoic acid? Retinoic acid is crucial for expecting mothers as it is an essential morphogen signaling molecule and major regulator of embryonic development. It is considered an essential nutrient because it cannot be produced through the human body, so it must be obtained through other sources which contain carotenoids. Carotenoids are antioxidant compounds found in both plants and animals that contain vitamin A. This vitamin A derivative is important for normal growth, eye health, and immune function. Luckily for us, it can be metabolized to retinoic acid upon consumption in the human body. When we first consume vitamin A, it is taken in and synthesized by the liver. It is stored in the liver as retinol ester and converted by two enzymes called acetyl-CoA, CoA, and LRAT. Upon entering the liver, retinol esters are then hydrolyzed by RBP to form the final product, which is retinol. Upon being created, retinol is released into the blood circulatory system as an endocrine signaler. It will travel through the bloodstream to embryonic cells. Within the bloodstream, it binds to transport proteins called transthyretin and retinol binding protein in order to form a protein complex. This protein complex will travel throughout the blood until it interacts with an embryonic cell. It must pass through the membrane by binding to the Q transmembrane receptor called STRA6. STRA6 will remove retinol from our transport protein complex and then assist retinol in moving across the cell membrane in order to gain access to the cytoplasm. Once within the embryonic stem cell, retinol will bind to CRBP and will be oxidized by ADH to form retinaldehyde, that is referred to by its common name as retinal. Retinol will be catalyzed by retinal dehydrogenase to form the final product, which is retinoic acid. Retinoic acid is then free to enter the cell nucleus and affect gene transcription and gene expression in one of two pathways. Our first pathway is when retinoic acid enters a target cell. This is a part of regular retinoic acid function. So the retinoic acid enters the embryonic cell, but in order to do so, it must bind to CRBP1 and 2. CRBP1 and 2 will then assist retinoic acid in entering the nucleus. Once in the nucleus, it will be deposited, where it can then attach to co-activators RAR and RXR attached to the rare protein subunit. This rare protein complex will process mRNA in order to activate DNA transcription onto the host genes, which retinoic acid is responsible for affecting. It regulates gene expression via downstream signaling. But in instances where retinoic acid enters a non-target cell, this is not ideal and we do not wish to have retinoic acid enter the nucleus. So instead, an enzyme called CYP26 will seek and target retinoic acid. CYP26 is responsible for degrading retinoic acid in order to regulate its function and ensure that there is not an excessive amount in the body. The reason for this is because retinoic acid is considered a tetragen. Tetragens are substances that cause malformation in embryos when found in excess. Since retinoic acid is concentration dependent, an overabundance of retinoic acid can lead to numerous growth defects. Retinoic acid has a morphogen gradient which subsequently activates host gene expression. Host proteins are transcription factors that regulate different brain regions and the segmentation of the neural tube into different vesicles. As a result, one of the more severe growth defects caused by the overabundance of retinoic acid is neural tube defects. Retinoic acid regulates almost all steps of neural crest induction, mitigation, proliferation, and differentiation. So when it malfunctions, it can cause the cranial neural crest to malform. As a result of this abnormal retinoic acid signaling and disrupting the formation of the cranial neural crest, there is a decreased amount of neural crest cell migration from the diencephalon to the mesencephalon. Because all of the cranial nerve cells tend to be centered in these two vesicles, there are less precursor cells in the resulting metencephalons and myelencephalons. These two regions of the brain located in the posterior compartment of the neurotube are crucial to the development of complex muscle coordination. The lack of this ability leads to a condition called midfacial cranial hypoplasia. This condition is characterized as a severe birth defect and is often seen as a deformation in the mandibular muscles of the face. It will often lead to lopsided jaws and mouth and a lack of bilateral symmetry in the eyes sometimes. This severe birth defect only stresses the need for the tight regulation of retinoic acid to ensure that there is just enough in the body to develop properly, but not so much that the body goes haywire. Thank you for watching.